So again, hi, I'm Hannah. I'm working as a technician for Dr. Angela Oliverio at Syracuse University. And today I'll be talking about elucidating the ecology, evolution, and genome biology of protists in extreme environments. So as we know from many of these talks so far, and from work in the field, extreme environments are well known as important study sites for the discovery of new lineages of life. And some examples of extreme environments are hydrothermal vents, geothermal springs, soda lakes, acid mine drainage, solar salterns, and cryosphere, just to name some of them. So thus far, these discoveries in extreme environments have largely taken place with bacteria and archaea. And from these discoveries, we've learned an immense amount about biological diversity and the limits of life from studying these groups in these extreme conditions. But we've barely scratched the surface of discovery when it comes to eukaryotes in extreme environments. So what about eukaryotes in extreme environments? This study is especially interesting based on some assumptions that eukaryotes cannot withstand the same conditions as bacteria and archaea, but many can, and we'll get into that. Here's a cartoon tree of eukaryotes with land plants, animals, and fungi boxed, and then everything else is microbial eukaryotes or protists, showing that protists make up the bulk of eukaryotic diversity. But when it comes to the study of eukaryotes in extreme environments, fungi have the most recognition in this area, whereas microbial eukaryotes are highly understudied in extreme environments, despite this phylogenetic diversity, as well as an increasing number of omics studies that are identifying diverse lineages of protists in surveys of extreme environments. I do want to point out that there has been some specific attention to protists in certain locations, such as deep sea hydrothermal vents, and also some early work in acid mine drainage, like in Rio Tinto. But overall, many locations have been largely overlooked, particularly geothermal springs, which I'll be telling you about today. So this lack of knowledge of protists in extreme environments has led us to synthesize existing data of protists in extreme conditions. And our perspective paper came out last week. So these data are available, where we compiled a big database, including data from culturing, amplicon sequencing, and more limited genome and transcriptome sequencing efforts, with the goal of identifying protist lineages of specific interest for further study in extreme conditions. And first, I'll be telling you about these culturing data. We identified over 50 cultured protist and fungal extremophiles, many of which have been cultured before the current decade, which gives us an idea of the maximum, minimum, and optimal conditions of protists across extreme temperatures, pH, and salinity, which we compared to fungi and then also bacteria and archaea, and found that protists can tolerate many extreme conditions that parallel fungi and even many that parallel bacteria and archaea. So first, looking at temperature, high temperatures were maybe one of the only conditions where bacteria and archaea can be much more extreme than eukaryotes, where bacteria and archaea are the only known hyperthermophiles, but many protists are true thermophiles. For example, the red alga Cyanoschidiosizon merle can withstand up to 60 degrees Celsius, which is the same as the highest recorded temperature for a fungus. Many amoeba are thermophiles as well. And then in terms of cold temperatures, many green algae are especially psychrophiles, living in snow and ice. In terms of halophiles, living high salt conditions. To put in perspective, seawater has the salinity of about 3.5%, but a flagellate halophile, Halocapiteria, can tolerate 10 times that level of salinity. Some other protist halophiles include other amoeba flagellates and some green algae. And protists are successful acidophiles and alkalophiles as well, with ciliates especially withstanding some high pH conditions, and red algae and an amoeba flagellate withstanding especially low pH conditions, as well as high temperature conditions at the same time for some of these, which makes them polyextremophiles, or withstanding multiple extremes at once. Beyond our culturing data, we also included molecular data from over 80 studies of eukaryotes across extreme conditions. And through this, we put together this phylogenetic tree of 18S RNA sequences from both environmental surveys and isolates. This is colored by, again, temperature, pH, and salinity. And through here, we can see some lineages with wide ranges of conditions that they tolerate, like the ciliates and chlorophytes, and then also some lineages that are found more specifically in certain conditions, like the oomycetes. Sequences from isolates are marked with the triangles on the outside of the tree, showing that many of these lineages have yet to be isolated. Many might be totally unknown protists, showing the importance of omics tools in the discovery of new lineages. And some of these are also deep branching lineages and might help resolve the eukaryotic tree of life. Also, the triangles of isolates that are black have published genomes versus the white ones do not, which shows an even further lack of data. And while there are many interesting takeaways from this tree, one is that amoeba may be prime targets for further study within extremophile protists. We found that two diverse groups of amoeba, lobosa within amoebozoa and heterolobosia within Discoba, are found especially in hot conditions, suggesting that there might be some adaptive advantage to the amoeboid form in high temperatures. Both of these groups have cultured isolates, but lack genome and transcriptome representation, except for Naegleria fowleri, which is a human parasite within Heterolobosia. For genomes that we did find that were published, we found some patterns of potential strategies for adaptation to extreme conditions, such as genome reduction, horizontal gene transfer, and genome expansion. But for the most part, we have no idea how most of these lineages have adapted to extreme environments. So we're interested especially in how diverse lineages, like these groups of amoeba, have adapted to these conditions. Some of these amoeba are also polyextremophiles, so it gives us an opportunity to study adaptation to multiple extremes at once. And we're interested in whether we'll find new genomic innovations or examples of convergent evolution between these multiple groups that are withstanding similar conditions. So this brings us to our current experimental work, where we're sequencing the genomes and transcriptomes of representative extremophilic amoeba and their closely related mesophilic relatives. So I just got back on Monday from a sampling trip to Lassen Volcanic National Park, which is about 4 hours north of the in-person part of this conference. Many of you are probably familiar with Yellowstone National Park, but Lassen actually hosts the largest geothermal lake in the US, Boiling Springs Lake, which is a flooded fumarole with sulfur-associated geochemistry. And it's also the prime target for an amoeba flagellate, Tetromytis thermocytophilus BSL, which was originally isolated and cultured by Gordon Wolf's lab almost a decade ago. I got in touch with Gordon when I was working on the synthesis I discussed, and also his collaborators. So we went out sampling and collected some water and sediment from Boiling Springs Lake, where we expect that amoeba flagellate, as well as two other sites, one that was high heat with low pH and one high heat and neutral, and took back over 15 liters of water on the plane with us, as well as soils. And we're already seeing amoeba in the initial enrichments we set up in the lab, which is very exciting. We are incubating now these water and soil samples at a range of high temperatures that we sampled from the park. And I've already amplified the genomes from 2 single cells, which we plan to send out for sequencing in the coming weeks. One of these amoeba likely includes Tetromytis thermocytophilus, which, as I mentioned, was previously isolated and cultured by Gordon Wolf's lab, but it was never genome sequenced. And we already have a mesophilic species of Tetromytis in culture in the lab for comparison. We're hoping to identify potential mechanisms of adaptation to the high heat and low pH conditions of Boiling Springs Lake using comparative genomics. We're very excited for our genome sequencing results. And based on what we learned from research in algae and fungi, which have higher proportion of published genomes, we hypothesized that genes involved in stress responses, heat tolerance, and heavy metal detoxification will be especially important to the success of amoeba in withstanding these extreme heat and acidic conditions. I'd like to thank the other members of the Oliverio lab, especially for helping process samples right now, Ken Stedman and the Extreme Virus Lab for taking us out sampling, and Gordon Wolf for an immense help as well. And I'll take any questions.
Okay, thank you, Hannah. Any questions from the right? You see any? Okay. All right, thanks again, Hannah. And I guess this ends the first part of the morning session.